Hello, everyone. Thank you for coming along to today's session. As Davor says, I'm Dr. Andy Holmes, and I work with himself, but also Kathy Gimlick and Dr. Chris O'Shea at the University of Birmingham. So it gives me great pleasure to introduce our speaker for today's session, Dr. Lili Wang. Lili is a very talented researcher whose work has been recently recognised as an Editor's Choice article published in JMCC, which I'm sure many of you will have read. Lili is currently a research instructor at Vanderbilt University Medical Center in the US. And before this, she obtained her bachelor's degree from Chengdu Technology University in China. Lili also did a master's in China at Southwest Jiaotong University. And then she moved to America, where she received her PhD from Auburn University in 2014. And after that, Lili joined Professor Bjorn Nolman's lab working on cardiac arrhythmias as a postdoctoral fellow. So I think we can all agree that Lili's research interests are particularly important and highly topical. And one of her key aims is to generate human iPSC-derived cardiac myocytes that have a more mature phenotype, so as to improve disease modeling and get to more effective drug screening. So this should be a really interesting talk. I'm sure there'll be many questions. Can I just ask you to please add these to the Q&A tab at the bottom of your screen? Okay, so without further ado, I'll hand over to our speaker today, Dr. Lili Wang.
Thanks. Let me share my screen. Thanks for Dr. Holmes' introduction, and also thanks to JMCC giving me this opportunity, this great opportunity to present our work in this webinar. So today I will talk about the steroid hormone T3 and the dexamethasone, just to change the expression of the potassium channels and increase the electrophysiology, like the maturations of the human-induced pluripotent stem cell derived the cardiomyocyte. Okay. So human-induced pluripotent stem cells, iPS cells, to generate these cells, our lab just did the skin biopsy from the healthy volunteer and collect the skin fibroblast, reprogram this fibroblast into the human iPS cells. So this human iPS cells can differentiate to the different type of the cells, like the neuron cells, endothelial cells, liver cells, and cardiomyocyte. So today I will focus on the cardiomyocyte. So in our lab, we use a small molecular method to do the cardiac differentiation. So we just split iPS cells as a monolayer and give the chair to drive the iPS cells into the mesoderm cells, and use IWR to drive the mesoderm cells into the cardiomyocyte. And we use the glucose-free medium to do the metabolic selection to increase the purity of the cardiomyocyte. And culture this myocyte for another 14 days to day 30. So we find that this day 30's iPS myocyte expressed the cardiac-specific markers, the sarcomere proteins, and the calcium handling related proteins. And also this myocyte has multiple cardiac ion channels. So this myocyte can generate very typical human action potentials. But this myocyte still function and structurally immature. So they're just like the fetal myocyte. They do not have any regular ultrastructure like the T-tubule network. And also they have the electrophysiological immaturations, like very depolarized resting member potential, very long action potential durations. So how can we improve the maturations of this iPS derived cardiomyocyte? So if we plate the iPS myocyte on the 1/60 diluted matrigel for 3 to 5 days, we can see the different morphology of this myocyte and with sarcomere disorganization and no contraction at all. So in 2015, we worked with Dr. Charles Holmes and Vanderbilt to develop a method called the maturation matrix method. This method, we still use the standard protocol to do the cardiac differentiation and get the day 13 myocyte. So we played this day 13 myocyte on the thick matrigel matrix. So this is undiluted matrigel for 3 to 5 days. What we find is we see a lot of the rod-shaped cardiomyocyte, and this myocyte with sarcomere alignment. And this method can generate a robust iPS contraction. So does this matrigel matrix method improve the electrophysiological functions of its iPS myocyte? To answer this question, we measured the action potentials and find the matrigel matrix method did not affect the resting member potential and action potential durations. But this method significantly increased the maximal upstroke velocities in the iPS-derived cardiomyocyte. As we know, this upstroke velocity is determined by sodium current. Nextly, we find is sodium current significantly increased in these matrix cells. So during the cardiac development, a lot of the expression of the ion channel just changes. For example, the pacemaker current IF. This IF is kind of the mixed sodium and the potassium inward current. And this channel can be activated and below the -40 or -45 millivolts. Acting these channels can trigger the spontaneous action potential and generating the pacemaking kind of activities. So in the DOS myocyte, this current only exists in the pacemaker cells. So during the cardiac maturation process, this current should gradually decrease to make the DOS cardiomyocyte. So inward rectifier potassium current IK1. So this current is maintained the selective resting member potential, and the outward current of IK1 also important for the late replications. Delayed rectifier potassium current IKR and IKS. These two currents are very important replacing the potassium current. So during the cardiac development, IK1, IKR, and IKS should increase or have highly expression to ensure the stable and constant action potentials in the DOS myocyte. So what happened to the matrigel matrix cells? We find that matrigel matrix does not change the pacemaker current IF and also has no effect on the IK1. So can we further improve the maturation of this iPS myocyte rapidly and constantly? So T3 is the steroid hormone. T3 and the dexamethasone is really important for the cardiac maturations. So we treat the day 16 myocyte with T3 and dexamethasone for 14 days. What happened? So we find that this treatment causes the cell hypertrophy, so the cell size is much bigger than vehicle-treated cells. Also, the T-tube density significantly increased in the T3 plus dex-treated cells, not T3 alone or dex alone. So how about the T-tube density compared to the human myocyte? We compare the human atrial myocyte and the ventricular myocyte. T3 plus dex-treated cells has higher T-tube densities than the human atrial myocyte, but has less than the human ventricular myocyte. So nextly, we look at the intracellular calcium handling and find that T3 plus dex treatment significantly increased the calcium kinetics in this myocyte. we repeat the same experiment, the calcium handling measurement with the SR block. And you can see after SR block, the calcium present significantly reduced in the T3 plus DEX treated cells. Also, the decay time constant was increased. So this tells us like the SR calcium make a great contribution to the calcium handling in T3 plus DEX treated cells. So T-tube T-tube can form the dienes to lead the L-type calcium current, L-type calcium channels close to the RYR2 on the SR. So since like T3 plus DEX treatment increase the T-tube densities, so how about the RYR activities and the structures? So we use the calcium sparks to determine the RYR activities and find that T3 plus DEX treatment significantly increased the calcium sparks frequency and the sparks mass. Also, in T3 plus DEX treatment, treated cells, we see a lot of the RYR clustering in these cells. So this data suggests T3 plus DEX treatment improves the structure and the function of RYR2 in the iPS-derived cardiomyocyte. So based on this information, we know this treatment is promoted the functional T-tube development and also improve the intracellular calcium handling in iPS-derived cardiomyocyte. But we do not know is like the effect of this treatment on the electrophysiological properties of this iPS myocyte. So Dr. Neil Gibson from the Esri Institute of Technology, he looked at the action potentials in the iPS-derived cardiomyocyte cell sheets. So what he did is he played the iPS myocyte on as the cell sheets and treated this cell sheet with T3 plus DEX or vehicle. And then he loaded the flow board to the cell sheets, recorded the action potentials like with the field stimulation. These are examples of vehicle-treated cell sheets and the T3 plus DEX-treated cell sheets. So what he find is, and the different pacing frequency, T3 plus DEX-treated cells has higher conduction velocities and shorter action potentials. So why? As we know, conduction velocity is determined by the inward current, which can cause the depolarization and also the gap junction channel collection 43. And since like the IK1, this current is important to maintain the negative the resting member potential. By setting up a more negative potential, the resting member potential, increased IK1 current can also enhance the availability of the sodium current. So we ask a question, does IK1 determine the conduction velocity of the cell sheets? To answer that, we inhibit IK1 by the barium chloride and find that 100 micromolar barium chloride can significantly reduce the conduction velocities. Also, IK1, the outward current of IK1 is important for the late repolarizations. So we find that barium chloride treatment significantly prolonged action potential durations in T3 plus DEX treated cells. But barium chloride, this treatment has no effect on the vehicle-treated cells. So as I mentioned before, collection 43, this gap junction channel is important for the conduction velocity. So Dr. Jeff Schiff's paper from the Vanderbilt, he looked at the protein expression of the collection 43 and find that T3 plus DEX treatment significantly increased the collection 43 expression. So that explains why T3 plus DEX treated cell sheets has higher conduction velocities than the vehicle. So nextly, we look at the spontaneous beating rate. Spontaneous beating rate is an important parameter to determine the maturation of the myocyte. As we know, the fetal myocyte has higher spontaneous beating rate, and the Dutch ventricular myocyte are still. So, as I mentioned before, our matrixual matrix method provides the physiological load to this myocyte and allows them to have the robust contraction. So I played the vehicle and the T3 plus dex treated myocyte on the matrixual matrix for 3 to 5 days and measure the spontaneous beating rate and the single cell level. Interestingly, we find the T3 plus dex treatment significantly reduced the spontaneous beating rate than the vehicle. Next, we look at the action potential and the single cell levels. What we find is T3 plus dex treatment causes a more negative resting member potential, increase the maximal upstroke velocity, shortens action potential durations. So our question, why T3 plus dex treatment improves the functions of this iPS-driven cardiomyocyte? Dr. Lili Wang from the UCLA, he used RNA sequence to compare the gene expression between vehicle and the T3 plus dex treated cells. So what he found is T3 plus dex treatment significantly changed the expression of the potassium channel related genes. And interestingly, we find like the casein G2, casein G12, these genes responsible for IK1 significantly increased. HCM4 responsible for IF, the pacemaker current, reduced. Casein H2 and casein E2 responsible for IKR significantly increased. Casein Q1 responsible for IKS also significantly increased. So our question is, are this gene expression change correlated with ion channel functions? To answer this, we use a patch clamp to measure the ion current and the single cell levels. What we find is this T3 plus dex treatment significantly increased the IK1 current, like 5-fold increase IK1 current. For IF, T3 plus dex treatment also 2-fold decreased the pacemaker current IF. So Dr. Yuko Wanda from the Vanderbilt, she looked at the rapidly delayed rectifier potassium current, IKR. So IKR is important replacing the potassium current. And Yuko just used like the E4032 to subtract the current and get IKR. This IKR current is present in every iPS-derived cardiomyocyte, and she finds that T3 plus dex treatment significantly increased the IKR. So she also looked at the slowly delayed rectifier potassium current IKS. So this IKS current is really tricky. Only less than 25% of the vehicle cells have this current, and about 75% of the T3 plus dex treated cells has this current. So with like HMR 1556, this is specific IKS blockers. She used this kind of the blocker to isolate the IKS current and find the T3 plus dex treatment significantly increased the IKS. So we find that the T3 plus dex treatment did not change the expression of SCN5A, but this treatment significantly increased the sodium current. So how does T3 plus dex treatment change the expression of this potassium channel or these ion channels? Previous study have shown that T3 and the dex DEX can regulate the transcription of the potassium channel genes by activating their nuclear receptors. So their nuclear receptor can activate the transcription factors to regulate the transcription, the gene expression. So we look at our RNA sequence data and find the THRA, THRB, that's for thyroid hormone receptor alpha and beta. NR3C1, that's for the GR receptors. Both also significantly increased in T3 plus DEX treated cells. Besides, the dexamethasone can also facilitate the potassium channel membrane trafficking by SGK, the serine protein kinase, and increase the potassium channel membrane expression. Our RNA sequence data show SGK1, SGK2 significantly increased in T3 plus DEX treated cells. So, in our studies, we find that if we treat the iPS-derived cardiomyocyte with T3 and DEX for 14 days, it can reduce the pacemaker current IF and reduce the spontaneous beating rate, can increase the IK1's inward rectifier potassium current, cause a more negative resting member potential. Increase IKR and IKS just lead a shorter action potential duration. Also increase IK1, increase the outward current of IK1, also contribute to a shorter action potential duration in T3 plus DEX treated cells. This treatment also increase the sodium current, lead to a faster maximal upstroke velocities. So we find that T3 plus DEX treatment just increase the electrophysiological maturations of its iPS myocyte in vitro. So these treated cells can be the better model for the human disease modeling and also for the drug screening in the future. For this study, I want to thank my mentor, Dr. Beyond Norman, and my colleagues. Also, I want to thank Dr. Neil Gibson and Dr. Ibin Wang's help. Thank you. Do you have any questions?
Okay, thank you, Lily, for a great talk there. So again, if you've got any questions, just pop them in the Q&A tab. So while people are doing that, perhaps I'll just start then. So just looking through the papers and the talk and thinking about this, I suppose the obvious question is, how did you come to look at T3 plus DEX? Was there a hypothesis behind, like, was it known that they regulate ion channels in the heart in particular, or in cardiac myocyte development?
Yeah, so this idea actually is coming from Dr. Christine Memory from the 2017 paper. So they use T3 and dexamethasone and IGF to treat their iPS-derived myocyte for 7 days, and they find that improves the mitochondrial function of this iPS myocyte, but they did not see any kind of ion channel expression change in their myocyte. So I just say, let's go ahead and try in our lab. We treat the cell longer, see this high dose of the hormone can stimulate the maturation of this iPS myocyte. So that's the thing. That's the reason why we use the 2 weeks treatment.
Yeah, brilliant. Thank you. So looking at the Q&A then, so we've got Guillaume Gilbert. Says, thanks for an interesting talk. In the T-tubule measurements, did you try on matrices only? As on the condition with T3 plus DEX without mattress, you see a similar tubular density as without hormones, similar for the SPARC data. In other words, what is the effect of the matrices themselves on tubule density and calcium sparks?
That's very interesting questions, and thanks. So we definitely look at the T-tube densities on the T3 plus DEX treated non-mature cells. So we find that the T-tube density is much less in these non-mature cells. We think the matrigel matrix provides a flexible substrate that's important for the T-tube formations. And also for the calcium sparks, we did not measure the calcium sparks, but we look at the RYR structure. So RYR did not form the clustering on the non-mature cells even after the T3 plus DEX treatment.
Brilliant. So we've got one here from Kim Helgren. Says, thank you for a great talk. Have you looked at mitochondrial function and or ATP production in these studies?
Yes, we look at the mitochondrial functions. So we published this data in 2000, in I think 2020 on Science. That also showed T3 plus DEX treatment significantly improve the mitochondrial functions and also improve the ATP production rate.
Okay, that's interesting, isn't it? Okay, so here's one from Max. Hi, Max. So it says, really great talk. Thank you. Have you tried the T3 plus DEX treatment on atrial specific hiPSC-CMs as well? Yeah, that's a question that I was going to ask as well.
Thanks. This is really a great question, the interesting question. So we our lab more interested in the ventricular like the myocyte. So and our differentiation protocol can produce more than 80% of the ventricular myocyte. So we did not try the atrial specific myocyte, but just overall this T3 plus DEX treatment is improve the maturation of this iPS-derived cardiomyocyte.
There you go, Matt. That's the next thing to try.
Yeah, I was going to say, I think that's definitely the next thing for us to try in Birmingham. So Tomoya Sakamoto says, thanks for a great talk. Have you tried to knock down T3 receptor and or the glucocorticoid receptor with a cocktail treatment to make sure that all the effects are caused by activation of these nuclear receptors? And two, why does only combination treatment work instead of either single T3 or DEX treatment on its own?
That's really great, and that's interesting questions. So we did not try knock down T3 and the DEX receptors, but that will be our future directions. Just to figure out the underlying mechanism of this T3 plus DEX treatment. So we try for the calcium for the T-tube densities, we tried to only treat the cells with T3 or DEX alone. We find that this only one growth hormone did not affect the T-tube density at all. Only just combine them together that can improve the T-tube kind of the density. And also I tried to measure IK1 with treated in iPS-derived cardiomyocyte treated with T3 alone or DEX alone. IK1 did not increase or did not change. So we think like combine the T3 and the dexamethasone together that are important for the iPS cardiomyocyte maturation. So both just upregulate the gene expression for the cardiac maturation, also improves like the protein expression or protein levels in this myocyte.
Yeah, it would be interesting to see where these two hormones actually converge, wouldn't it? It's an interesting question. Okay, so we've got sorry,
Jump in, if you don't mind.
Yeah.
I always have this really bad habit. I apologize. But you tried titrate. Is this a titratable effect? You know, is there a concentration dependency here? Have you kind of worked this out? Is there a plateau that you're reaching here with your treatment, or could that be the reason why, you know, a single hormone sort of exposure doesn't work as well because you haven't maybe maximally activated these receptors? I know they're separate receptors, but
Yeah, that's a really interesting question. So in these studies, we use very high dose of the T3 and the DEX. You can notice that we use 100 nanomolar T3 and 1 micromolar dexamethasone. That's really high concentration to maximize their effect. So we did not try the different concentration. Maybe lower concentration still can have the same effect.
But is there I don't know what your output would be, but is there a way of testing, what your response to these things is? Are you at the top of your curve? Are you saturating your response, or are you somewhere in the middle? Whereabouts are you on the kind of response curve? Is there a way of assessing that? Because I know you say it's a high hormone level, but I don't know what that means. Maybe you've only activated 20% of your receptors with that high hormone.
Yeah, that's really interesting. So we never measured the saturation, kind of the collectives of these receptors, so we don't know. But compared to these hormone levels in the human, we think we use very high dose, so that can maximally activate these receptors.
So what are they in the humans?
I think it's about it's much higher, as I remember.
Lower, you mean?
Yeah, in human it's much lower than this treatment. So during the development, the T3 level and the dexamethasone level is much lower.
Thank you.
Okay, so Almudena Velasco says, thank you for a great talk, Dr. Wang. Have you checked if the treatment with T3 and DEX changed the expression levels of key calcium handling proteins, such as the ryanodine receptor, SERCA, or NCX?
Thanks for this question. So I think we checked the gene expression for this calcium handling proteins. For RYR and the SERCA did not change after T3 plus DEX treatment. Gene expression did not change does not mean the protein levels also not changing in these cells. We measure the intracellular calcium handling and also measure the calcium flux through these channels. Just like calcium sparks, we find the T3 plus DEX treatment just increase the calcium flux. So that means T3 plus DEX treatment just increase the function of these proteins.
Yeah, it's interesting, isn't it? I mean, coming back to more sort of general expression levels, I saw that you did the RNA sequencing. But in the paper as well, we just saw the ion channel gene expression, but with the RNA sequencing, were you able to sort of look more broadly to see whether there were any other changes in transcription factor expression that might be driving some of these other changes in the ion channel expression? I don't know if you're able to look at that.
That's interesting question. So Dr. Lili Wang he's from the UCLA. He's doing that project right now, just to compare the transcription levels of this kind of the regular these genes and see how the T3 plus DEX treatment help the ion channel gene expression.
Yeah, okay, that sounds really interesting. Yeah, I look forward to seeing that then. I mean, definitely that was something I thought of when I was going through the paper was what's the mechanism, the exact mechanism which is underlining these changes in ion channel function. Okay, so moving on back to the questions then. So Rosanna Bassani says, thank you, great talk and results. Have you investigated sodium channel isoforms?
Isoforms, we did not. We look at the total sodium current.
Yeah. What about beta subunits with the something that could be quite important as well, the beta subunit expression? Did you look at that possibly?
And no, sorry, we did not look into that. But we will study that probably for the future directions.
Yeah, I mean, yeah, just looking at the inactivation curves, they're pretty consistent actually. So maybe there weren't any changes in the beta, but could confirm that. Okay, then. So I'll just end then with one final question, and just more of a sort of general feeling really about where we are with the field. So obviously the treatments have some pretty major effects between sort of treatment and vehicle groups. But how close do you think we are to resembling the electrical phenotype of, say, primary human cardiac myocytes? Is there a way to go, or do you think we're nearly there now?
So even after T3 plus dex treatment, these cells, compared to the dust myocyte, the cells still immature. It can tell by the cell size. The cell size is still much smaller than the human myocyte. And also this ion channel expression is much lower. So I think in the future, we will work more on this maturation method, see how can we just further improve their maturations. But our T3 plus dex treatment definitely significantly improve their structure and the functional maturation of this iPS myocyte.
Yeah, brilliant. So still a way to go, agree. Yeah, but this is definitely a step in the right direction, isn't it? Which is really promising for the field. Okay, so I just want to finish by thanking our speaker again, Dr. Lili Wang, for a fantastic talk today. And also just want to remind everyone to maybe sign up for the ISHR in Berlin if you can. Okay, thank you very much for today's session.
Thank you, everyone. Can I just say on my behalf as well, really nice talk. Andy, thank you for chairing, and audience, thank you for tuning in. The talk will be on YouTube as well, put up shortly. So thank you. See you soon.